She was actually she had some friends in the hills around there who had some. So I'm gonna call to order the 1246 regular meeting of the Entomological Society of Washington. And I'm going to start out my name is Don Weber. I'm the current president. And I'm going to start out by apologizing for it not being in person, or at least the option of in person. This was due to some late notice on the part of our host, the Smithsonian Museum of Natural History, and they're doing a generator test, which could, well, it could not only disrupt our meeting, but they said you were not welcome. So I pretty much made it virtual. Welcome, everybody. This is our first regular meeting since our annual potluck banquet. Could you please mute yourself? Or Alan, you can mute them all. Since when was I muted, Lourdes?
Maybe 15 seconds, 30 seconds.
Okay, I don't know
That
how
was me.
but whatever. So as I was saying, this is our first regular meeting since our potluck banquet on June 8th. And I want to thank again our president-elect Andrew Jansen for organizing that banquet and for others who contributed. And it was a great success. And we had an excellent talk on cicadas. And I believe that means that our last regular meeting was May of 2024, and that Secretary Gary Hevel still has to report on the minutes from that meeting. Is that correct, Gary?
Yes, it is.
Okay, I'll turn it over to you then.
Okay. At 7 p.m. on the 2nd of May, 2024, the 1245th regular meeting of the Entomological Society of Washington was called to order by President Don Weber in room WD 33 of the National Museum of Natural History. At the meeting were 12 members, 5 guests, and 14 virtual attendees. Recording Secretary Gary Hevel read the minutes of the April meeting, followed by an approval by attending members. President Weber noted that the intended speaker for the annual banquet is not available, but President-elect Andrew Jansen is completing details for a replacement. Lourdes Chamorro noted that there are plans for a shuttle to the event from the Greenbelt Metro Station. Membership and Communications Officer Elizabeth Young announced that Yvonne Turner, Jeremy Frank, and Liz Jones are officially society members as of this evening. Program Chair Alan Norbaum briefly noted that he found leaf mines in golden ragwort plants and also found flies of the genus Straußia in 2 species of sunflower plants. Program Chair Alan Norbaum introduced the speaker of the evening, Dr. Karen Berghart, an assistant professor in the entomology department at the University of Maryland College Park and a research associate at the Smithsonian Environmental Research Center. She specializes in understanding plant and insect interactions in human-modified landscapes, ranging from suburban yards to abandoned agricultural fields to managed forests. An introductory slide image was of the morning glory prominent, a species that does not feed on morning glory, and was an example of faltered information in our knowledge of plant and animal relationships. For evaluation of plant and animal relationships, extensive plantings of trees were undertaken with some 18,000 trees of 116 native trees. Volunteers and citizen scientists were utilized in this large research program. Experiments are growing and ongoing. Visual sampling for caterpillars were conducted with 4 minutes per tree. A wealth of information was gained and provided data for plot comparison. Caterpillar numbers and compositions were gained throughout the plot. Cicadas were studied because they cause most damage to trees. Caterpillars were found to survive best on trees in diverse plots. This vast ongoing research is providing many insights into forest compositions. And that's what I have. Any changes, errors, problems, insults?
Hearing none, call for the approval of the minutes. All those in favor say aye.
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 Any
Aye.
opposed? Thank you, Gary.
Okay.
I'm going to report on two major things, and I don't think it's going to take a major amount of time. I want to report first that the proceedings, our journal, the Proceedings of the Entomological Society of Washington, is in very good shape due to the diligence and hard work of our relatively new editor-in-chief, Jerry Cook. Jerry, are you on the line? Okay, he's not. But we also concluded a new deal with Bio One, which allows for open access, but we still get the same fees from Bio One, or maybe more. And in any case, it's a good deal. And I was going to ask Jerry to comment on it, but he can do that next meeting. And the executive committee has approved that deal. And the other good news about the journal is it's getting up to date. We're only one issue behind. And thanks to Jerry's good work on that. Remember, if you're a member, you can publish in this with no page charges. And your membership includes already electronic access. This new deal, which gives open access, is taking effect in January of 2026. Any questions on that? I think Matt could answer them if I can. All right, hearing none, I'm going to go on to the next big item, which is a revision of the bylaws, which the executive committee has been working on quite a while. And I want to thank now for the long and hard work of Abby Kula, our treasurer, working with the lawyer and constructing these new bylaws. And I'm just going to summarize them and some of the reasons why they're happening. First of all, some research, I think on both Abby's and the lawyer's part, revealed that the Entomological Society of Washington continues as an unincorporated association. And that jeopardizes our status to manage our funds and to take legitimate charitable contributions. So the purpose of the bylaws, in part, is to convert into a District of Columbia nonprofit corporation. Any changes in the bylaws, by the way, need to be presented to you, the membership, those of you who are members, at this meeting, and all of these new bylaws, as well as the markup showing the changes, are already posted online at our website. If you have any trouble in accessing those and you remember trying to look at them, please let us know. And what the plan is to present that tonight and to have you review it, and then in the November 7th meeting, 2024, to vote on approving those. So basically, this does allow proper management of our available funds for investment and our tax and a legitimate tax exempt status to encourage contributions by members and others to the society. I'll just go through a few more points of what it does. Nothing shocking, I assure you. We're clarifying that we're a member-governed society and keeping the language that the society is organized and operated exclusively for scientific and educational purposes. We're stating we have simplified the membership categories, which consist of regular, student, life, emeritus, and honorary. and those are all defined, and they're not different from what they were before. We're explicitly allowing electronic participation for official meetings. That wasn't explicitly excluded, but we want no doubt about that we can hold virtual and hybrid meetings. We're adding to the executive committee and the leadership a post entitled Website Administrator. And that's going to be added to officers and describes its duties, which are pretty straightforward on what you would expect. They manage the website. We're clarifying that the past president, who's currently Matt Buffington, is also an officer. That's easy. And consistent with the new status as an incorporated 501c3 corporation, reserve funds can be managed with investment accounts, and the treasurer does that with the advice of the finance committee. It's not too different than what we do now, except that it's clear now we have legitimate nonprofit status and can manage those with no second guessing. So I think that pretty much summarizes that. Abby's on the meeting and can answer any detailed questions that anyone has at this point. I'm not hearing any unless you're muted.
And I think we also sort of changed the makeup of the finance committee. Like,
it changes the makeup of the finance committee.
yeah.
That didn't make it to my top 7, Alan.
Okay. Well,
but you're
probably nobody's going to complain.
Yeah. Okay. All right. Hearing no more discussion of that, I'm going to remind the membership, and we need all of you who are on here now and probably a few more to show up and vote on this. And I think we will have an in-person meeting November 7th. That's our next meeting, the first Thursday of each month. And I urge you to review those. I can assure you that the vote amongst the executive committee was unanimous, and these changes are, in my opinion and the opinion of the leadership, a very good thing. Abby, did you want to comment at all?
I don't really have any comments, except that I hope everybody will review the 2 versions, the marked up version and the final edited version, and come to the meeting on November 7th so we can vote.
And I want to thank Abby again for really far beyond the call of duty here. I think Matt has been working on this in prior presidents as well.
For sure, Alan.
Come to fruition. And okay, and Alan. So next order of business is, do we have any new members to announce, or after that, do we have visitors to the meeting tonight?
Perhaps, Dawn, you might want to let people know that there is a vacancy of the membership secretary, which might be an important point to make.
There are 2 vacancies, although Alyssa Seaman just did volunteer to be nominated for the membership and communication secretary. As far as I know, Andrew, we have no nominations so far for president-elect.
Correct.
We will be voting on the slate is it November? Yes. We will be voting on the bylaws and the slate of leadership in November. So that will be ready soon. Do we have any visitors who want to raise their hands and tell us who you are and anything else you want to know? Well, we need to know your name.
Excuse me, Don.
Yeah.
Oh, We usually announce new members, but I guess we don't know since the membership chair is vacant. But I did exchange messages today with Evan Economo. He's the chair of the entomology department at the University of Maryland. He told me that he just signed up, so can at least announce his name.
Good to know, Alan. Thank you.
Yep.
And we also have Joella Killian. Joella, do you want to, as a visitor, do you want to tell us about you? And
Oh, hi everyone. I'm a retired entomologist, tree fruit entomologist. I worked with Doug Pfeiffer at Virginia Tech, and I just have a great interest in tonight's topic. Thank you for letting me join.
well, thank you for attending.
My pleasure.
I'm an old friend of Doug's, so.
Okay.
Anyone else? I saw Joella in the chat, and I see a bunch of icons in the chat. Sue, and also for those of you who want to access the bylaws later, Lourdes has posted the link in the chat. It's pretty straightforward. It's entsocwash.org about the society, and you're going to find it pretty easily.
Don,
Yes.
since I did agree to be acting membership secretary, and I do have access to some of the back end of the website, there's another name, and they identify themselves as a new member, and their name is Julie Craves from Ann Arbor, Michigan. And I'll see if I can go down the list a little bit here, and maybe I have a few more names. Okay.
Thank you. Let's see. I wanted to call for any unfinished business or other new business items. Anyone? At this point, if we were in person, and we can also do it here virtually, does anyone have any presentations of notes, books, or exhibition of specimens? Going once, going twice. Good. If no one has any more to say at this point, I'm going to allow Alan to introduce our speaker for this evening. Alan, are you ready?
Yep. Hi, everybody. Before I introduce our speaker, I just want to tell Don we have 35 attendees tonight. A good turnout. And our speaker in November will be Armando Rosario Lebron, who works for the National Identification Service, USDA APHIS, and his topic is going to be the invasive threat of imported invertebrate pets.
Not pets.
Pets, yes. Somebody's commenting, yay, Armando. Okay, so our speaker tonight is Yelena Pacheco. She received her bachelor's and master's at Brigham Young University, and she did her PhD at the University of Georgia. She is currently a USDA postdoc in the Systematic Entomology Lab, working on aphid and chrysomelid phylogenetics. But tonight she's going to talk to us about fireflies, which I think we all agree is a really cool topic. So thank you, Yelena.
Thank you. Thanks everyone for logging on. I'm bummed I didn't get to meet you all in person, but I'm still excited to get to share some of my dissertation research with you guys. So starting here, I'd like to kind of give you guys just a little introduction to myself and give some context for my interest in entomology. But when I started undergrad years ago, I didn't have the desire to be an entomologist. I wanted to be a surgeon. In fact, I didn't even know that you could study insect evolution. I thought the only people who worked on insects were, like people in IPM or, vector control. So during my second year at BYU, I started working in an evolutionary biology lab, and I had the opportunity to do research, but also go and do field work, and that's where I instantly fell in love with entomology. And I was fascinated by the diversity I saw while collecting, and of course, running around in the jungle was a really good plus too. And so I kind of went from there. Long story short, I ended up ditching the dream to go to med school and decided to go to grad school instead. And now I've already alluded to this, but one of my major research interests is evolution, and I'm really curious on how organisms and lineages have changed over time. And these changes result in the diversity we see in organisms today, and I really love being able to observe and document this diversity. In particular, I'm interested in morphological diversity of insects and how structures have changed over time, and fireflies are a great group to study these topics, allowing me to address some really interesting questions about insect evolution. So fireflies, while a smaller group of insects with only about 2,500 species, are still extremely diverse. As you can see here, they have different shapes and sizes of their eyes. They also have, sorry, let me see if I can get the pointer. All right, I don't think there's a laser pointer, but different shapes of their eyes, their antenna, and even their light organ, which is the yellow or white tissue you see there at the base of their abdomen or the end of their abdomen. And that's where they produce light. So I'm sure most of you are familiar with fireflies, seeing them on warm summer nights, but let me just give you a little more formal introduction to them. I think a lot of you will already know this, but in case anybody doesn't, fireflies are a family of beetle, the family Lampyridae. And like I've mentioned already, they're very morphologically diverse. So of course we see really big differences in their antennae between different species and then their eye size. And in addition to that, we see a lot of differences between species in female adult forms. So you have female fireflies that are fully winged and look very similar to males, but in some species you have brachypterous females whose elytra are really shortened or reduced, and then you even have neotenous females that can look like larvae to some extent. And so fireflies or Lampyridae obviously get their name from their ability to produce light or bioluminescence. And all fireflies bioluminesce as larvae, and they use this, it's thought that they use this as larvae to ward against predators and let them know of their chemical defenses. But in the adult stage, they use bioluminescence primarily as a sexual signal to attract and locate mates. However, not all adults produce light. So there are some species that do not produce light at all in the adult stage. So we even see diversity in firefly sexual signaling. So nocturnal fireflies use bioluminescence to attract and find mates. But like I said, some species lack light organs as adults, and they actually use pheromones to find their mates. So in the nocturnal species, males will actively fly around at night, flashing and waiting for a female to respond to them, and then they'll fly down to the female and attempt to mate. And then in diurnal species, females are again sedentary and they'll release pheromone plumes, and males are searching around looking for those pheromone plumes to locate a female. And then within bioluminescent signaling, we see even more diversity. So some species flash while others use continuous glows. And this is just an example of a few species and their different flash patterns. And these species-specific patterns help males and females within the same species identify each other. So as I've already mentioned, there's these two different signaling methods within fireflies. So nocturnal fireflies rely on their eyes to detect their bioluminescent signals, while diurnal species rely primarily on their antennae. And we know from previous work from Stinger Hall et al. That nocturnal fireflies have much larger eyes relative to their body size, while diurnal pheromone signaling species have much larger antenna relative to body size. So this really shows the importance of both structures relative to which sexual signal method that they're using. Now, bringing this back to my research interests, when I started at UGA, one of my questions was, what other morphological differences do we see between nocturnal and diurnal females? So this kind of brings us to the first research part of the talk, but during my PhD, I decided to look much more closely at the firefly antennae. So insects, their antennae serve as the insect antennae serves as a major sensory organ for insects, collecting information about their environment. This can include, chemical signals, temperature, humidity, a wide range of things. And insects' antennae are made up of multiple segments. Fireflies tend to have 11 segmented antennae. There are some exceptions in certain species. But these antennae or antennal segments can be greatly modified. So you see here, we have some typical filiform, like cylindrical segments. You have serrate antennae that are blade-like, and then all variations of branching antennae. And these are all examples of firefly antennae. Now, when I said we'd be looking more closely at antennae, I meant a lot more closely. So this is a picture of an individual antennal segment taken with a scanning electron microscope. If you look at the bottom here, we're at about 900 times zoom. And so what's really interesting about these antennal segments is they're covered in all of these hair-like structures, which are actually part of the insect cuticle, and they're called cincilla. And then when we look even more closely at the cincilla, we see additional diversity on top of that. In fact, there are actually 3 different types of cincilla here. You can see the long, wispy ones, some short, kind of stubby ones, and then some medium length ones. And I'll talk more about those in a bit. But cincilla are used to detect environmental stimuli. Most insect scintilla can be grouped into 4 main functions, including touch or pressure, chemical detection, temperature, or humidity receptors. And so for my dissertation, I focused primarily on the chemoreceptors that might be used for pheromone detection. And within these different functional groups, you tend to see certain morphological groups associated with specific functions. So for example, Chiatica and Capaniform mechanoreceptors typically are associated with mechanoreceptors, and then Trichoid, Basiconica, and Coloconica are typically associated with chemoreceptors. And while I was interested in solely the external morphology at this point of cincilla, I just wanted to give you a brief insight on how the cincilla works. So here we have a picture of a olfactory cincilla. You can see that there are pores here that would allow chemicals to enter the cincilla and excite these nerve dendrites, and then the signals carried on through the cells and onto the nervous system. One of the things that I wanted to know about firefly cincilla was how many different types there were on the antennae, and then do all species have the same type of cincilla, or do different species have different morphologies? So to answer this question, I looked at the antennal cincilla of 7 different firefly species by sampling both taxonomic diversity and signal diversity. So I have 4 subfamilies sampled here, both bioluminescent and pheromone signaling species, and then I have 3 males and 3 females from each species. In the majority of our taxon sampling, males and females of the same species look basically the same. They're the fully winged adult form. However, our Faustus species we sampled has neotenous females. This means that females look very similar to larvae. This species also is our only glowing species that I sampled, and is hypothesized to use possibly a combination of pheromone and bioluminescence during mate search. And then just a little fun fact, this is, I don't believe this is the picture of the exact species, but this is another species in the genus. But the species of Phallus that we used in this was just recently described by our very own Alicia Hobson. So, Alicia, I have the names updated in some of these slides, but not all of them, but they are updated in the manuscript, just so you know. So to determine which Cincilla morphotypes were present in each species, sorry, I used scanning electron microscopy images to look at those microscopic structures of the antennal cincilla. First, I removed the antenna from the head, I cleaned them, I dried them, and prepared them for gold coating, and I took over 2,000 images of firefly antennae with a SEM at the Georgia Electron Microscopy Lab. And so all of the photos that you see of antenna and cincilla from here on are photos that I took. So when I went through my thousands of images and identified the different types of cincilla, I found 14 unique morphotypes. And then I categorized these cincilla into functional groups based on morphological work by Schneider and then comparison within other beetle groups as well. So as I begin to talk about these morphotypes, you'll see that they have been given some names. And so these names are based on a scheme introduced by Faucho's 2020 paper that synonymized cincilla morphotypes among elitarian beetles. And so we decided to just go with that to kind of make things easier for future comparisons across Elateroid families. So here you can see our 3 mechanoreceptors. And you can see, so in this first picture, we have our cincilla base that comes directly out of the cuticle. And this is a long cincilla with grooves. And these are known as C1 cincilla. They're fairly commonly found throughout other beetle groups. And then we have these bome cincilla, which are smooth and they come to a sharp point. They're very short relative to the other mechanoreceptors that we see, and these tend to be located on the most proximal end of the antennal segment. And then we have our disc cincilla. So these we found in much lower numbers, which I'll talk about in a second, but their names are pretty self-explanatory in their morphological description. Now, these are the 9 chemocincilla I found. They vary in the shape of the base and then the peg that comes out. So all these on top have a fairly dome-shaped base with a short or medium-length peg, while the ones at the bottom have very different bases, and some even lack a base completely, like our trichoid cincilla. Now, these last 2 are actually, one's a suspected thermoreceptor. So we suspect that just because it's been something with a very similar morphology was tested in other beetle species. And then the second one are cone cincilla, and right now we don't have a good enough hypothesis to place them in a functional group. So this table shows all the different types of cincilla I found for each species. So we have 7 species, but plus this Luciola cruciata at the end here, and that is the first species to ever have its antennal cincilla documented by Iwasaki. So here you can see like the C1 cincilla are found in all species of firefly. Most of the mechanoreceptors are found throughout all species with a couple exceptions. But then when we look at the chemoreceptors, you see a lot of diversity in terms of some being found in multiple species, some being found in just one species, like our B12 and 13 or B10 and B11. So once I knew what types of cincilla were on the firefly antennae, I wanted to identify other potential differences between nocturnal and diurnal species, and detecting which ones used pheromone signals for mate search. So I had two hypotheses. First, that diurnal species would have more sensilla than nocturnal species. Obviously, the diurnal species are the ones using pheromones. We don't know if nocturnal species use pheromones, but we would assume that they primarily use their visual cues. And then the second hypothesis that males would have more sensilla than females because, again, males are the ones actively searching for those pheromones that the females are emitting. So to quantify these, I counted over 140,000 sensilla by hand, and then separated those numbers out into their respective morphotypes. I also measured the area of the antennal surface and then used the length of the firefly pronotum for a proxy of body size. In addition to knowing how many sensilla were present, I wanted to know how fireflies increase their ability to detect pheromones. So do they increase the size of their antenna, allowing more room for sensilla, or do they increase the density of sensilla in a given area? To test this, I also calculated the sensilla density for each specimen by dividing the total number of sensilla by the total surface area of the antenna. So this is just a few of the tests that we run for our hypotheses, but first we looked at the total number of sensilla and found on the firefly antennae, and we found that diurnal sensilla have more overall chemo and mechanoreceptors than nocturnal species, but they don't have any differences in their density. So this suggests that they do increase sensitivity by increasing their antennal size rather than packing more sensilla tightly on a given surface area. And then I also found that males had significantly more sensilla than females, but equal density, which again is supports that mating behavior, or supported by the mating behavior where males are actively searching around for female pheromones. But to better understand pheromone signaling, we need to focus just on the chemosensilla. So again, these are our chemosensilla, and to test the differences in counts and densities, we looked at the total number of chemosensilla just because our sample size wasn't big enough at this point to look at individual sensilla morphotypes. So we found that males have significantly more chemosensilla than females, and that they also pack them more densely on their antenna, which again supports our hypothesis of the males needing the increased sensitivity to be able to better find the females. And then we found that diurnal females have the same number of sensilla as, nocturnal females. So we don't see any differences in the number of sensilla between females, which, supports our hypothesis that really the sensitivity or the need to detect pheromones is, reserved for the males. But we did find that diurnal males had significantly more chemosensilla than nocturnal males, again showing that pheromone detection is more important to diurnal species. While I showed the importance of chemosensilla in diurnal fireflies, and more specifically diurnal males, I still wanted to see if I could identify potential pheromone sensilla. So traditionally, this is done using known pheromone compounds and electrophysiology recordings, but we don't know the pheromone compounds used by most firefly species in general. In fact, we only know the pheromones for one of the species in this study. So I wanted to see if there were other ways that we could at least find potential pheromone sensilla for future testing, like the best way to narrow down our first physiology tests if in the future. So looking back at this table, we can see there are no sensilla morphotypes found exclusively in diurnal species and not in nocturnal species. So this indicates that there's not one universal pheromone sensilla morphotype, and that pheromone sensilla are likely species specific. To identify potential pheromone sensilla, I identified two criteria that would suggest morphotypes might be used for pheromone detection. So the first is that males will have more of a specific morphotype than conspecific females. And then the second is that the morphotype has pores. And so as a reminder, pores allow the volatile chemicals to enter the sensilla and stimulate the nervous system. So here's just an example on the right of what pores can look like in one of our B3 morphotypes. So again, our sample size is not big enough at this point to be confident or have enough statistical power to do comparisons, but I think this is just a really interesting observation looking forward. But we have in our Photinus caruscus species, so this is a diurnal species, we see that males have quite a greater number of our B1 sensilla than females. And then in a recent paper published in 2022, it was actually tested with electrophysiology and found that B1 is the pheromone detecting sensilla for caruscus. And then if we look at Lucidota punctata, so they have two potential candidates, our B10 and our B11. Males have quite a bit more, almost a little less than double than females. And then we do find pores present in this type. And then really interesting in our B11, they have almost 100 times more than females. But we weren't able to detect pores at this point on those sensilla. And then finally in our Pyropygia, again, a diurnal species, males have a much larger number of the B3 sensilla that do have pores than females. So again, these are just kind of preliminary hypotheses and like suggestions that if somebody wanted to physiology testings, these might be the first ones to look at. Let's see. Yeah, I kind of already went through all of this, but these are just, again, pictures of the types. So our B3 sensilla, which have pores present, and B10 and 11. So that brings us to the end of this first part. Just in summary, I found 14 different firefly sensilla morphotypes. 10 of them are new to fireflies and 2 are new to beetles overall. I showed that diurnal fireflies, specifically males, invest more in chemosensilla than nocturnal species. And then we identified 4 potential pheromone sensilla candidates. So I actually am okay, like, well, okay, future directions. So we obviously want to increase our sample size, look at a greater diversity of species and sample sensilla with different antennal shapes. So right now we only have filiform and serrate antennae. So looking at some of those branched antennal structures would be great too. And so what I was gonna say is I would be happy to take any questions right now about this part. If there are any, feel free to speak up. If not, I'll go ahead and move on to the next.
Well, Yelena, thank you. I know you're not through, but just looking at the central picture there, I kind of got this contrast with feathery antennae, pheromone, no light, no light organ, and obviously that's not true from the one picture. So is it conceivable that some of them have both pheromones and also use bioluminescent signaling for courtship?
Yeah, definitely. So Phausis is one of the genera that we think might do that because so those are the blue ghost fireflies. So their bioluminescent signal is like fairly faint compared to what we think of when we see like the Photinus or the Photur species. So that's one I'm kind of blanking on. There's about like 4 other genera that we think have really strong reasoning to believe that they are doing both.
And the Phausis is is that whole genus Neoteny in the females?
That I don't quite recall. I know there's more than one species that has neotenous within that genus. So, but I can't say for sure that they all are.
Yelena, I could ask a question to Alan. When you were showing the images of the antennae, it almost looked like there were scales or something on the antennae, or is that just like some kind of surface sculpturing? But that was kind of
Yeah, no, I think that is, let's see if I have a good picture. I know what you're talking about. Well, I guess
yeah,
here
that
this
one
what
showed it.
we're yeah. No, I think it's just more micro sculpturing, and it does tend to differ based on species. So like some species, if I remember correctly, like the Photurus, that's pretty smooth relative to some of these other ones that they do look scale-like, but I don't think they're actual scales in like the term of like they're removable. They're just micro structuring. So.
Yeah. Is there a like a phylogenetic pattern in whether they're diurnal or nocturnal, or do they switch back and forth in different lineages?
Yeah, let me I have that at the very end here. Let me jump to this. So really, as we have now we haven't done any formal analyses on it, but if you look down here, the nigricans, Cruscus, and Pyralis, right? These are the most closely related. You do see like the B1, maybe in the B3, but not strong enough. I don't think we have enough diversity at this point to really say for sure. But there are some that completely go like this B2, or these X1. They're seem to be completely uninfluenced by phylogeny.
So you're saying that you can't speciate any of these with the sensilla?
So you mean like it's, sorry, I'm not quite sure what you mean by speciate them in the sensilla.
Okay, well, let's say with Photurus, the bane of my existence, it's hard to tell what species you have. I mean, you can look at them and you can look at their flight pattern, but I'm just wondering if there's anything other than DNA, maybe, that would separate the different species, say, within Photurus.
Yeah, so I mean, unfortunately, we've only been able to look at one species of Photurus at this point, and when we look over here, they share the same, like their two chemoreceptors, oops, sorry, are found in other species. So at this point, no. And I would suspect that's pretty consistent. I think that'd be a pretty drastic change between species to see these different like completely unique morphotypes per species. So I don't have any images of them here, but if we look to, sorry, it keeps jumping back. Like the Lucidota punctata. So I looked at another Lucidota species and they also have B10, and those are like really unique. We haven't seen those in any other beetle species, at least that's been documented that I could find in the literature. So I would assume that like it would maybe be helpful for genera at the lowest level, but probably not species, unfortunately.
I just thought I'd ask. Thank you.
Oh, yeah. All right. I guess I'll look at the chat, but last call for questions on this part, and then we'll move on. If I can get the chat open. It keeps disappearing from me. Oh, thank you, Alicia. Well, there's only 2 of the 13 Phausis species have neotenous females. I knew she would know. Okay. All right, so moving on to a little bit different vein, but still talking about Firefly evolution. One of the projects I was tasked with and interested in as part of my dissertation was to reconstruct a worldwide firefly phylogeny. And so this has been a great collaboration over many years with multiple people working on this project, multiple people contributing specimens, and without them, it really wouldn't be possible. But I already stated, fireflies, I think that number from 2,200, probably closer to 2,500, like I said earlier. And then they have 11 described subfamilies. And then in the most recent large-scale molecular phylogeny of fireflies, 8 subfamilies were represented. So our goal was to increase our diversity on this. So my phylogeny that I worked on, we have 339 species, and currently right now 9 of 11 subfamilies represented. We do want to add, we have the potential to add 10. Just got to work on the data there a little bit more. But what's really cool about this is like the Chespereutini are represented for the first time in a large-scale molecular analysis, and we really increased our neotropical taxa as well. So for this phylogeny, we used anchored hybrid enrichment loci. So these target highly conserved regions flanked by less conserved regions. So this allows us to collect data that's useful to resolve both deep and shallow nodes on the phylogeny. And our target probe set consisted of 516 loci. Once we had those loci assembled, we used Piper to do that. We removed any loci that had, or removed any taxa that had less than 250 loci, just to minimize our amount of missing data. And then that gives us 342 taxa. Then we removed any loci that had less than 50% taxon coverage. So we are down to 481. Then we removed any sites with less than 50% taxon coverage. And we did coalescent tree reconstruction and also looked at gene tree discordance. But our final data set consisted of 458 loci with 342 taxa. So this cladogram, the one on the left, represents the newest phylogeny of Lamprididae, and then the one on the right is the Martin et al. Phylogeny. And these are just cladograms of the subfamily relationships. So obviously, some of the subfamily relationships have changed. Most notably, the Odotratini are recovered as non-monophyletic. And now I'll just take you through this topology in a little bit more detail. So in addition to Odotratini being recovered at the base of Lampyrids, it's non-monophyletic, but that's not completely surprising considering the Martin study, it only had 2 taxa represented. And so here we have 19. So in the previous phylogeny, we didn't really have a big enough sample size to be confident in the monophyly of that subfamily. But that's definitely something that we want to look further into, look for morphological characters to determine if that result is supported by the morphology as well. And then the Chespereutini, this is one of the ones that I was most excited about to see where it's placed, because they're just like a really weird looking firefly, but very cool. They were recovered as sister to Polyclassis, but I will note that this is only represented by a single species. So it's a pretty small group, and we just don't have a lot of samples, but we're really grateful for the one that we had at this point. And then Polyclassis is a monotypic genus, meaning, right, most of you know that it only has one species. So which subfamily it belongs to is really been questionable up until this point. But Polyclassis' placement as sister to Chespereutini is not entirely supported, considering the clade's placement in the relationship to the Teratini. But again, these specimens, so our Polyclassis or Teratini, and then our Chesperetini, they're only supported by 2 to 3 taxa as well. So, there's always a possibility of some sort of like long branch attraction or something going on there. So that's something we're still working on before we get this paper out. And then another interesting group, Vesta, which has both New World and Old World taxa. They were recovered within the Photorhini. It's really interesting because most of the Old World taxa are usually Luciolini or Lamperini. But this is something that Sarah Rivera and Luis de Silveira are working out at Western Carolina University. So keep an eye out for Sarah's thesis. And I think she'll have some more answers there for us. But now that we have this big, robust phylogeny of fireflies, we're able to address some really exciting questions about how firefly sexual signaling has evolved. But first, I'll just give you a background on the history of firefly sexual signals. So we know that the common ancestor of fireflies used pheromones for mate search. However, bioluminescence has evolved independently multiple times in this family, and there have also been multiple losses of bioluminescence. To determine how these signals have changed over time, I coded sexual signal for as many species on the tree that I could find information for, and the character states for these sexual signals included pheromones, flashes, which are very short bursts of light, and with periods of darkness in between, or glows, which are continuous outputs of light. And then I ran a parsimony ancestral state reconstruction in Mesquite using our new phylogeny. So how do we determine the number of independent origins for character evolution? One way to do this is with a parsimony analysis. So we count the minimum number of times a character state changes. However, sometimes there are multiple explanations. In this example, in the evolution of the green circles, we can see on the left the possibility of green circles evolving twice and being lost once for a total of 3 changes. But sorry. Or we have green circles could have evolved independently 3 times for, again, a total of 3 changes. So because of scenarios like this, we sometimes see multiple estimates for the number of times a character has evolved independently. So I found, in this case, a possibility of 5 to 8 independent origins of bioluminescence, 5 to 9 origins of the flashing signal, and 6 origins of glowing. Now I'll take you through some interesting highlights of signal evolution. In most cases, I found that flashes evolved from a pheromone ancestor, like the example in the right. But there was also one instance where flashes actually evolved from a glowing ancestor. And then additionally, I found that glows evolved from both pheromone and flashing ancestors. In addition to the evolution of bioluminescence, I was interested in the evolution of how structures that produce the flashes or the light organs have changed. So here you can see our different types of light organs. These are modified from Branham et al., but we have the entire ventrites being covered up. You have a bar where there's only light organ in the center. You have a single spot, and then you have paired spots. So again, I coded the species on our phylogeny that we had information for, and I found quite a big, found that light organ shape was really variable and likely could very plastic across species. So we found 10 to 12 independent origins of light organs occupying the entire ventrite, 15 to 17 origins of the bar shape, 10 to 11 of the single spot, and again, 10 to 11 of the paired spot shape. So some of the interesting findings for this test include identifying the ancestral state of the Luciolini light organs as occupying the entire ventrite. And we also see both bar and paired spots within the same subfamily. And then our 4 light organ shapes were found in the subfamily Lampreyni and Photorhini. So these findings again suggest that light organ shape can change relatively rapidly between species. Now, finally, I wanted to see if light organ size could be a predictor of signal type. So do flashing fireflies have larger or smaller light organs than glowing species? First, I calculated the phylogenetic signal of both absolute light organ size and relative light organ size. The absolute light organ size is a measure of the total light organ area, while the relative size is a measure of the light organ proportionate to the total ventrite size. Analyzing the relative light organ size can give us an idea if the area of the ventrite that the light organ occupies is influenced by phylogeny. And then phylogenetic signal is a measure of how much a character is influenced by phylogenetic relationships. In this case, I used Pagel's lambda. A lambda value of 0 indicates that the character is not influenced by phylogeny at all, while a lambda value of 1 indicates that the character is completely influenced by phylogeny. So using my coalescent phylogeny and a measurement of light organ size, I was able to determine that relative light organ size was strongly influenced by phylogeny, but also light organ size was not. Now that we know phylogeny strongly influences light organ size, we need to account for character variation due to phylogeny in order to determine if signal type can be predicted by light organ size. So to account for this variation, I use phylogenetic generalized least squares analysis, using both, again, my phylogeny and then measurements of eye, antenna, and light organs. I tested models for both light organ relative and absolute size. And I found that neither relative or absolute light organ area were significantly correlated with signal type, aka flashes or glowing. So unfortunately, this means that like if you had a firefly, say like in a museum collection that you have no life history on, at least we don't have the data in our model to indicate whether or not this firefly might be a flasher or a glower based on its total or relative light organ size. To summarize my findings for this portion, there are plenty of opportunities for taxonomic work in land purity. So I'm really excited about that for future firefly workers. And then it really highlights the need for these taxonomic experts. And then additionally, bioluminescence has evolved independently multiple times in fireflies, suggesting that signal type can occur relatively, or signal type can change relatively quickly on an evolutionary timescale, as well as light organ shape can change relatively rapidly. And then finally, relative light organ size is strongly influenced by phylogeny, making more closely related species have similar light organ sizes. So just as a final reminder, during this talk, we talked about the great diversity in firefly and Temple cincilla. We are working on the most comprehensive phylogeny of fireflies to date, which will hopefully be a really great tool in the future for people wanting to look at specific lineages. And then we trace the evolutionary history of firefly sexual signaling and light organ shape. And so with that, I would just like to thank my my advisor for my PhD, Dr. Sanger Hall, everybody in our lab, my past committee members. It's kind of cool that I'm doing this talk today because about a week ago today I defended my dissertation, so it's been almost exactly a year. So it was nice to kind of remind myself of all the cool things I got to do during that time. And of course, again, anybody who sent myself, UGA, Mark Branham, Seth Bybee, specimens, it was super useful. This great phylogeny wouldn't be possible without that. And now I will take any questions about either part of the talk. And thanks for listening.
Thanks,
Hey,
Yelena.
Lena, I got a question.
Yeah.
So I was really awesome work. What an amazing system to be able to work on. I just had a couple questions about your phylogenetic results. The first one was which probe set did you use? Is that something that you guys developed, or did you use that was a probe set that was already developed for a different group of beetles, or what? And a
Yes.
kind of a go-along question is what sort of support values did you see across the nodes in your tree at various depths in your tree?
Yeah, so we use the Firefly probe set, so it's the same one in the Martin phylogeny, and it was developed off of the big beetle probe set, but just specialized for fireflies. And then support values, it was not, so by my standards, it was not great for some of our deeper nodes and our subfamilies. I don't remember quite off the top of my head, but that's something that I am working on. So we use Filuche for this, which doesn't give you as much information in your assembly at like those more or those less conserved ends of your probes. So one thing that I'm working, sorry, did I say high? We used Hyde Piper. I don't know what I just said, but we used Hyde Piper. So one thing that we're working on is using Filuche, or there's some other pipelines out there to see if we can get more data that might either support some of those deeper nodes or some of the more shallow nodes. But I will say, I guess our most shallow nodes tend to be fairly well supported. It's really just like the backbone that, again, with most other phylogenies has low support. But I would say ranging from like 40 to 75%.
Okay, cool. Thanks.
Hi, Yelena. This is Matt here. Congratulations. Great talk. Um, the Going back to the phylogeny, I noticed that Vesta appeared to be paraphyletic. You mentioned it was Old World and New World. Is one of those clades, the Old World, going to be like actually Vesta, and the other one's going to be a new genus, or how is that going to be handled?
Yes, I believe so. So that's something that a master's student at Western Carolina is working on, but I believe that is their plan. So we produce like a whole bunch more. So that's only like a sample of the Vesta we have. So she's gonna have a lot more sequences, a lot more species represented.
Yelena, this is Alan. So are lampreyrids like way more diverse in the tropics, like most
Yes.
insect groups? So that's why you're excited to get all the neotropical stuff.
Yeah, and so we have quite a few new species in our sample that Luis is trying to he is working on describing, and then at UGA my old advisor Katrine and then Joe McHugh, if you know him, they have 2 PhD students and they're looking for another master's student, and their whole project is doing South American firefly taxonomy, so that's really exciting.
Good. It looks like, sorry, my dog keeps barking. It looks like there's a lot of switching back and forth in like at least the types of signaling organs they have. Like, is there a lot of switching back and forth between whether they use light for signaling at all? Like, or do you, and like what would be the selective pressure for that? You have any idea? Are they going from nocturnal to diurnal and back and forth?
Yeah, so the ancestral state is thought to be diurnal. So there's been tons of switches to, or like relatively, there's been lots of switches to no light to using light. And then there's just a few switches relatively to, again, losing light. So yeah, so our best hypotheses for adults using light versus not using light in terms of how that pressure influence speciation would be like habitat competition, right? So obviously if there's too many species in a certain area, they're all emitting light signals. If their signals aren't diverse enough, perhaps that there's selective pressure on those that might subsequently lose light. I think we see that too with, right, you see different activity times with fireflies. So you have those that are active more, at dusk versus those later at night. And again, I think that's just so that you're not, that's, how am I trying to say this? That's a way to reduce like possible misinterpretation of somebody else's light signal is signaling at those different times, if that makes sense.
Yelena, I had one more question. So, I can see from this picture two different colors. I've heard reference to blue ghost, and I've seen in Amazonia at least some very orange ones as opposed to the greenish yellow that we're familiar with in North America at least. Can you comment at all on the colors and whether they might have some value in distinguishing or looking at the evolutionary phenomenon of different spectra?
Yeah, so what is really interesting, one of the other grad students that was in Katrina's lab with me, she looked at color between different populations of the same species. So we know of like populations of Photinus in the south that I believe are more yellow in color, while populations in like the northeast, like Pennsylvania, New York, New Jersey, they're more green in color. The interesting thing about that is like their luciferase is the same. This is the sequence for luciferase is the same. So what she looked at was like, and she's not quite finished with like the conclusion, but was different screening pigments. But that does seem to be like a way for them to an extra way to distinguish between them. I think we just don't know quite enough about what is actually causing the different colors. I feel like I kind of like forgot the last part of your question, but like how that has to do with evolution or
Well, yeah, there's some that seem so different, like the blue ghost and some of the orangey yellow that it seems like it must be a different um, production rather than what you referred to, which is a whole nother aspect of.
yes. Okay, so yeah, I guess now I don't, of course, now I don't remember. I believe, oh, I'm trying to remember if those that are extremely different, they have different luciferase, like they have key differences in their luciferase sequences that make them different. And I believe they do, if I remember correctly.
So I had a comment. Here in the Northeast, we do have a fairly orange Photinus scintillans, and it's pretty orange. It's like 578 nanometers maybe. And you can look up the paper by Bruce Branchini, because I collected for him and he looked at luciferase and luciferin, and you can, see what causes it. But I don't know how different it is. I mean, it's a Photinus. Nobody's changed the species or the genus or anything. So that's just the point. And we also have Pyractomina that somebody could look at, Pyractomina borealis. And that one has a sort of an amber flash, and it's not like the flashes of our, well, it's actually central US flashes. It's a different color, sort of amber. Just saying.
Yeah, those would definitely be all great ones to look at. The paper I'm thinking about by Sarah Lauer, I believe they just looked at those um a few species of phallus with those southern and northern populations. Or sorry, not phallus, photinus with southern and northern populations, and like I said, they didn't find differences. But I feel like some of the labs over in Japan have looked at it as well, in terms of the evolution of luciferase.
Yeah, they also took the genetics, the genes for this Photinus scintillans and stuck them in E. coli, and they present with the same colors. I mean, so it seems to be totally genetic and not something else.
Yeah.
That's Thank you.
Yeah, thank you.
Thanks very much, Yelena. I guess there's no other questions. We can turn it back over to Don. Thanks so much for speaking to
Thank
us
you.
tonight. It's a really cool area.
Cool. Thank you. Yeah, thanks for inviting me.
Well, thanks again, Yelena, and if we want to unmute and give you a hand, I'd love to hear that.
And
Um, Thanks again. That's fascinating. I wanted to call on Lourdes before we close the meeting. I guess he's done a little research on our new membership trends. You might summarize that, Lourdes.
Yes, thank you. Just kind of quickly, I filtered the website to find new members since May up until now, and we've had 14 new members at least. One didn't specify whether they were new or not, but so I'll just quickly say their names as the membership secretary usually does. So Evan Economo, Julie Craves, Pamela Zader, David Hewitt, Lauren Widner, Thomas Skarlinski, but I'm not sure if they're new or not. Derek Rosenberger, Andrew Meads, Michael Caterino, Austin Fife, Robert Finn, Tina Litwack, Alan Cabrero, and Hector Gonzalez Hernandez. And this last one from Mexico. And a few of them are new students. So welcome to all the new members since May.
Thanks, Lourdes. Any more anyone has to say, or I'll adjourn the meeting. Thanks, everybody, and apologies again that we had to go virtual, and we'll see you at the first Thursday in November, which is the 7th, and we'll hope to see many members there because we have our bylaws and leadership elections and an exciting talk by Armando. Thanks, everybody. Meeting adjourned.
Thanks, Don.
Bye, thanks.